Today we are going to observe the cells of onion under microscope. So for this we need slide, cover slip, stain, and a piece of onion, some water, and a needle. So first we have to cut this onion because we don't need this much. Okay. After that there will be a thin membrane of onion. We have to take out that thin membrane, and then we have to put it in the stain so that the cell wall will take the color and it will be clearly visible under the microscope. Okay, so we will place it in the stain and we have to wait for 2 to 3 minutes. Okay, okay, now the time is over. We will take the membrane and we have to wash it. Okay, so first I'll wash it clearly, and I have washed it. The water is also red in color. Now we have to place that membrane onto the clean slide. Now we have to spread it. So the piece is we don't need this much, so that's why we are cutting the extra piece. We need only a small piece of this membrane. Okay, so we have removed the extra pieces. Now we will add some water onto that membrane. After adding some water, we have to place the cover slip onto this. We have to place it gently so that there will be no air bubble. If you see any air bubble, then you have to press it so that all the bubbles will come out. As you can see, there are 2 air bubbles. So like this we can, by pressing it gently, we will solve this problem. Okay, so now the slide is ready for observing under microscope. Now see, first I have to keep it some other places. As you can see, this is the microscope, and first I observe it under first we are going to observe the cells under 10x. Okay, now see how it looks under the microscope. It is 10x, so it will be not that much big. You can see all the cells, and inside the cells, means inside the rectangular boxes, you can see some round objects, okay, like bubbles, and they are called vacuoles. It is very difficult to see something under the microscope using phone. See, it is clearly visible. You can see in each cell you can see some round balloon like or air bubble like structure. Those are vacuoles. Okay, and cell wall is also clearly visible. Now we are going to observe it under 40x. For that, again I have to place it tripod.